right, I think the mic's good. I think we're in here. All right, what's going on, guys? Erickmed here. In this video, I'm going to show you guys how I tackle these biological and biochemical foundations of living system passages in the MCAT. Okay, so we're going to be doing the MCAT official prep section bank passage. We're going to do a passage in the section bank. So basically, when you buy all the materials on the AMC website, you get all of these things here. You get the question packs, practice exams, sample tests, official guide to the MCAT, all these stuff here. And the section bank, these questions are known to be of a higher difficulty than everything else. Okay, it's been known on Reddit. Everyone talks about it. This is definitely, and when I did it, I'm like, okay, these are like definitely more complex, harder passages and questions than the practice test. So these are really good sources of material to practice on. So we're going to be going ahead and doing one of these passages. We'll do the first one. Okay, we're going to be doing this passage. So I recommend, guys, I mean, I don't know if I'm allowed to even like show you guys this stuff because I know you have to pay for this. So we'll see how long this video stays up for before it gets taken down. But okay, I want you guys to read this, pause the video, read this, read everything here. Look at this table here. This is the first question. Okay, this is the second question. That's the second question. Pause it, whatever, go through it. Third question. Fourth question. Fifth question. And this is sixth question. I believe there's six only. Yeah, there's six only in this one. So go ahead, pause it for the questions, pause it to read this. And then as soon as you're done, you got your answers, write them down, and then resume this video because I'm going to be breaking down this passage. And hopefully it'll make a lot more sense. Or hopefully you get everything right too. But, okay, let's begin. So, looking at this passage right now, let's say if I'm taking the MCAT and I just get this, I'm like, okay, I'm looking at this, and it doesn't seem that bad. But you never know. So, we're going to read this here. Okay, blood flow to the brain is regulated by changes in arterial vessel diameter in response to intravascular pressure, a process called the myogenic response. The myogenic response allows cerebral resistant vessels to achieve a state of partial constriction from which they can dilate or further constrict to alter blood flow. Okay, just reading that right now, I've never heard of the myogenic response ever, but I did hear before and in my content review and studying that the pressure of blood, the blood flow, can be altered when you dilate or constrict your blood vessels. So usually when your blood vessels are dilated, blood flow goes faster and blood pressure usually increases. And when your blood vessels dilate, blood flow usually would slow down. So I do remember that. So this is kind of making sense here. So now I'll go to the second paragraph. Vascular smooth muscle contraction is initiated by cell membrane depolarization, which opens voltage-gated calcium channels. Calcium ions bind the systolic protein calmodulin, which activates an enzyme that phosphorylates amino acid residue 19 of the myosin light chain. Phosphorylation of LC20 is required to activate the myosin head, which binds to actin. This myosin-actin interaction forms the basis of the cross-bridge cycle, which causes muscle contraction, as in skeletal muscle. Okay. So we're talking about the blood flow to the brain, those vessels, how they can dilate and restrict and constrict in order to alter blood flow. Reading this paragraph, you do have to be familiar with how muscle contraction works. You do have to be familiar with the actin, the myosin head, how the myosin head binds to actin, the exposed actin sites when troponin is basically on the muscle, and then once calcium binds to troponin, it exposes the actin sites for the myosin head to bind onto. So you have to know that background information. But they give it to you here. So if you're not even like perfect with that background information, that content knowledge that you should have reviewed, you can still, I believe, answer these questions. So anything here, they gave us calmodulin, which is a protein. I've never heard of that protein before, but it's fine. We do know, okay, right here, some people would get tripped up. Vascular smooth muscle contraction initiated by cell membrane depolarization, which opens voltage-gated calcium channels. Okay, you have to know this is very common in the MCAT in a lot of passages, like what depolarization is, what these voltage-gated calcium channels are. You have to know a lot of that stuff. And it also tells us here that, oh, phosphorylation is needed to activate a myosin head. Okay, so we know it's not too bad. Dynamic reorganization of the actin cytoskeleton occurs during vascular smooth muscle contraction and relaxation. Okay, makes sense. During actin polymerization, monomers of globular actin, G-actin, polymerize into filamentous actin, F-actin. Actin depolymerization can be induced by the drug letrunsulin B. Okay, this is stuff that I've never seen before. And you're not supposed to see this before. You're not supposed to study and realize, like, oh, during actin polymerization, G-actin polymerizes into F-actin. Like, no, that wasn't in any of the Kaplan review books. I don't remember reading that in any of my classes in undergrad. So this is new stuff here. So they threw this new stuff here. Also, be aware that they threw this thing here. Literally, it tells us, okay, G-actin goes into F-actin. And they threw this fact here. Oh, actin depolymerization can be induced by the drug letrunsulin B. So what I would do is, if I have time, of course, I would highlight this. Just highlight this real quick. Because this is something they threw in there. And usually when people are in a rush, they're reading through this and they look at the question, they forget about this, that this is needed to answer the question. So last paragraph, to examine changes. Okay, always right before this table here, the paragraph before the table is usually more complex. Like 9 times out of 10 out of 10, 10 times out of 10. Usually they'll give you a chart, table, graph, a Western blot analysis, SDS page, like a bunch of chromatography, anything, separation techniques. The paragraph before is usually a little more complicated. So take your time for this part, because this is what literally helps you understand the experiment that they do in all these passages. So to examine changes that occur in vascular smooth muscle during the monogenic response, researchers exposed isolated rat middle cerebral arteries to increasing internal pressure to mimic intravascular changes that would occur in vivo. Arterial diameter and the amounts of phosphorylated LC20, phosphorylated calponin. Phosphorylation of calponin reduces its inhibition of the actin-activated myosin MGAT pace, and G-actin were determined in response to a 3-step pressurization from 10 to 80 to 120 millimeters of mercury, whatever, pressure. Okay. So now usually when you're reading this, time is of value. So you're limited in time. So you read this, understand what's happening here. Read this part. This is very important here, because this tells you what everything's about. Changes in RMCA, so the rat middle cerebral artery diameter and protein levels in response to artificially adjusted internal pressure. So this is the pressure here, internal pressure. This is artificially adjusted. So it goes from 10 to 80 to 120. Okay, when this pressure is adjusted, they look at, okay, pressure adjusted, what happens from that? Arterial diameter, it decreases. Phosphorylation of LC20 increases. This increases. This decreases, it looks like. So to save time, read through this. The table, don't even really look at it. Just jump straight to the question. So do this, read the title, and then boom, jump right into the question. Don't try to look at the table and try to analyze it, or the graph, or whatever chromatography technique they give you. Don't try to analyze that before you jump into the question. Go ahead, read the passage, the words. Don't look at the tables and graphs. Jump into the question first, because that will save you a bunch of time. Because usually, like this question right here, I read all this and it's like, based on the information in the passage, which type of enzyme is most likely to suppress CBC? Okay, so this is a pseudo-discrete kind of question. Okay, so you have to know your enzymes here. Which type of enzyme is most likely to suppress CBC? Okay, CBC, well, I'm pretty sure it was a cross-bridge cycle. I remember reading about that. And I think it was like, oh, phosphorylation of this does something. Okay, so let's read real quick here. Phosphorylation of LC20 is required to activate the myosin head, which binds to actin. This myosin-actin interaction forms the base of the cross-bridge cycle, which causes muscle contraction as in skeletal muscle. Okay. So When LC20 gets phosphorylated, the myosin head gets activated, binds to actin. This literally forms the basis of the cross-bridge cycle, which causes muscle contraction. So, phosphorylation forms the basis of the CBC, causes muscle contraction. Which type of enzyme is like to suppress the CBC, to suppress muscle contraction? Well, if phosphorylation activates it, and phosphorylation is adding a phosphate group, removing a phosphate group would most likely suppress it. So phosphorylase, this adds a phosphate group. Kinase adds a phosphate group. Phosphatase removes a phosphate group. And synthase, I forgot what synthase does. But I know that it removes a phosphate group, so I'm going to go with C. See, that was very easy. This question was very easy. Okay, yeah, I got it right. Okay, next question. Which statement regarding VSM function and cytoskeletal dynamics is best supported by the data in Table 1? Okay, so here's Table 1. Now we look at it because it literally says best supported by the data in Table 1. Okay, vasoconstriction is associated with an increase in the ratio of F-actin to G-actin. Well, vasoconstriction is constricting the blood vessel. When you're constricting the blood vessel, that diameter, I'm pretty sure it goes down because you're constricting it. So the diameter goes from 225, it goes to 175. So this is constricted. So this is associated with an increase in the ratio of F-actin to G-actin. Well, as you can see here, G-actin goes down. I remember in the passage something about F-actin. It was like, F-actin, I'm pretty sure when we have G-actin polymerized, it goes into, right here. So during actin polymerization, monomers of globular actin polymerize into filamentous actin. So the G becomes F during this polymerization. Okay, so G becomes F. The less G we have, the more F we have, because all that G is becoming F. That G-actin is becoming F-actin. So we're constricting here, and the G-actin amount goes down. It goes from 1 to 0.3 to 0.1. So when we're constricting, we get less G, increase in the ratio of F-actin to G-actin. When G goes down, F goes up. So this does make sense. Looks like A is the correct answer. But let's check B, C, and D. F-actin levels decrease as phosphorylation of LC20 increases. Okay. Remember, when G-actin goes down, F-actin goes up. So here's phosphorylated LC20. As this gets phosphorylated and these levels increase, G-actin decreases. So F-actin increases. This is wrong because it's saying that F-actin levels decrease as phosphorylation of LC20 increases. That's wrong. F-actin levels increase as phosphorylated LC20 increases. Vasodilation does not affect phosphorylation levels of calponin. Vasodilation is the opposite of constriction, so the diameter would increase. So as we vasodilate, does not affect phosphorylation levels of calponin. Okay, as we dilate here, this does not get affected. This stays at 18, goes to 18 as we vasodilate here. However, as we vasodilate from 193 to 225, 18 to 7, there is a difference here. This is affected. So this is incorrect. Arterial diameter reduction is always dependent upon increased calponin phosphorylation. Okay, here's a key thing here. Whenever you see answer choices that say that are very extreme, like, oh, never, always, like those words, they're very extreme. So that's a strong conclusion you're doing here. Arterial diameter reduction is always dependent upon increased calponin phosphorylation. It's a very strong statement, and strong statements require strong evidence. So this needs to have some strong evidence here. If it does not have strong evidence, if I'm iffy about it, I'm going to chop the axe and get rid of this answer. So let's see. Arterial diameter reduction is always dependent upon increased calponent phosphorylation. So we're reducing the diameter. This does increase, okay. Reducing the diameter, this stays the same. It's basically like the other answer choice here, but the opposite. It's not always. It's only from 80 to 120 it is, but not always. We're not always increasing the calponent phosphorylation. We're only increasing it from here to here, but not from here to here. So this has to be wrong. So I'm going to go with A as the correct answer. And yeah, that's right. Okay, next question. Compared to control, our MCA held at an internal pressure of 120 mmHg. Application of latrosulin B to RMCA held at 120 will most likely result in what? Okay, see, right now, through these questions, we're already understanding the passage. I already understand this passage completely. Okay, I understand this table. I understand what's going on here. So these questions, they're easy. So, okay, control RMCA. So the control, they say, is the 121. So this is the control. So compared to this, when we add latrosulin B, it'll most likely result in what? Okay, what the hell is latrosulin B? Well, we know that depolymerization can be induced by drug latrosulin B. So B depolymerizes actin. If D depolymerizes actin, that means we're going to have less F actin and more G actin. Okay, so you should, by reading this, you should already know like the answer. Okay, you should already have a thought in your answer. So let's check which adds up to my answer before I even read the answers. Okay, increase phosphorylated LC20 levels. No, because no. This drug stops it. This drug inhibits actin. It induces actin depolymerization, this drug. Why would it result in increased phosphorylated LC20 levels? Doesn't make sense. So I'm going to go with the X here. Decreased arterial diameter. Arterial diameter. Listen, when we get F actin, this means we are contracting, we are polymerization. We're losing F actin, and we're gaining G actin. So we're going this way. We're gaining G actin here. So this 120 here, this is telling us that we're decreasing arterial diameter. We're losing G actin. As we can see from this table, we're increasing the diameter. We're not decreasing the diameter from this way to this way. Remember, we're increasing the G actin amount. This is increasing the diameter, but this answer choice says that we're decreasing the diameter. So this is wrong. Decreased F actin levels. Yeah, I said that before I even answered this question. So I'm pretty sure it's C. Increased phosphorylated calponent levels. Wrong. Again, we're increasing the actin amount. So we're going this way. Increased phosphorylated calponent levels. No. This stays the same here, and then it goes down. So this is wrong. So our answer is C. Okay, next question. According to information in the passage, which statement best describes the function of the myogenic response in response to stimulation of the sympathetic nervous system? Okay, you got to know what your sympathetic nervous system is. You got to know what it does. So I know that sympathetic nervous system is your fight or flight response. I know that in context of the blood flow and all this stuff, I know that the sympathetic nervous system constricts your blood vessels. So blood flow increases to your muscles, to your whatever. So you're more alert, you can act quicker, all this stuff during your fight or flight response. So we'll see what answer choice adds up with that. So the myogenic response equalizes blood pressure in cerebral resistant arteries in the aorta. Equalizes blood pressure in cerebral resistant arteries in the aorta. I do not remember aorta being mentioned once here. This is aorta. This does not according to the information in the passage. There is no information in this passage about the aorta, so I'm going to X this out. Moderates blood flow to the brain under high pressure. I believe, yeah. Yeah, this is the myogenic response is basically like, okay, like we dilate or we constrict to alter this blood flow. So this seems like a good answer. So it seems like a good answer, but I want to read the other ones first. Enables cerebral resistant arteries to locally vasodilate. Yeah, they did mention dilation here. From which, see right here, from which they can dilate or further constrict to alter blood flow. This answer choice makes sense. Redirects blood circulating in the brain to organs in the abdominal cavity. Okay, you did not talk about the abdominal cavity here. This is according to information in the passage. There is no information in this passage about redirecting the blood to the abdominal cavity or aorta that was like mentioned here. Like what? So I'm typing the X here. Okay, now we're left with 2. This usually happens a lot during the MCAT. But just relax. You can do this. Big dog, big bark, no bite. Let's die. Okay, so if you're like right here, okay, what the heck is the answer here? Because they're both kind of like the same thing. This passage mentions that the cerebral resistant vessels to achieve a state of partial constriction from which they can dilate or further constrict to alter blood flow. This literally says it. It literally says it can dilate, vasodilate. And this says moderates blood flow to the brain under high pressure. By dilating and constricting, that's literally you're moderating the blood flow to the brain. However, when this happens, reread the question. And careful not to like choose something and then, reason your way into it being the right answer. Stick to the evidence. According to the information in the passage, which statement best describes the function of the myogenic response? So this whole thing here, in response to stimulation of the sympathetic nervous system. So, in response to stimulation of the sympathetic nervous system. This means sympathetic nervous system goes first, and then we have the myogenic response here. The sympathetic nervous system, I know it's going to create some high pressure because it does constrict blood flow. So I'm leaning towards this. Enable cerebral. This is true. It does do this. It does, from which they can dilate or further constrict. It does do this. However, this is correct and this is correct. However, this is a better answer because it's more specific. It's saying this is in response to the sympathetic nervous system. This is just saying what it does. But the question asking in response to the sympathetic nervous system. So it's B. I'm pretty sure it's B. See, the correct answer is B. Again, the question asked in response to the sympathetic nervous system. This answer choice is correct. This answer choice is correct if the information in the passage. However, this answer choice is more correct because it answers the question better. Okay, so again, I'll explain this again if you didn't really quite understand it. The question asked, what's the function of the MR in response to stimulation of the sympathetic nervous system? Okay, MR in response to the sympathetic nervous system. The sympathetic nervous system creates a high pressure, constricts blood flow. So in response to that, the MR, myogenic response, moderates the blood flow to the brain. Does the MR enable cerebral resistant arteries to locally vasodilate? Yes, it does. However, the question's asking in response to the sympathetic nervous system. This is more specific. This is not. So usually specific answer choices, they're usually correct more of the time, if it makes sense. So the correct answer is B. All right, here we have kind of like a pseudo-discrete question here. I can already tell by looking at it. Okay, the polymerization of which structural component was analyzed in the experiment describing the passage? We're talking about actin here. I don't even need to read the passage over. Actin, I already know that it's made out of microfilaments. Okay, actin is not made of microtubules. That's tubulin. Intermediate filaments is made from actin and tubulin. Thick filaments, I don't even know what that's made out of, but this is a pseudo-discrete. I know it's microfilaments. You should know this from your content review. Okay. Last one. is the likely structure of the amino acid found at position 19 of LC20? Okay, I remember saying that phosphorylation of LC20 is required to activate the myosin head. Phosphorylation of LC20. You should know your amino acids like the back of your hand for the MCAT, especially in this section. And also, you're probably going to see some amino acids in the Chem and Phys section as well. So you should already know here. Phosphorylation of an amino acid. Which amino acids can get phosphorylated? The ones with OH groups are usually more likely to be phosphorylated because they have the hydroxyl group. So which one has a hydroxyl group here? This one. Bam. Oh, and that's pretty much it. Okay, this passage. Honestly, this passage wasn't that bad. Maybe it's because we broke it down, but this is a section bank, so these are known to be a little more difficult. But as you can see, as we broke this down, okay, as we knew what we knew, and we knew what we don't know, as we noticed, okay, like this is not that bad. This is a big dog, but no bark. I mean, big dog, big bark, but no bite. We were able to answer this because it literally says phosphorylation is required to activate. And this literally says phosphatase, which removes a phosphate group. Okay, so this is like, duh. Again, right here, this is very important to know, okay? Polymerization. G-actin polymerizes to F-actin. That means we lose G-actin and we gain F-actin. That's exactly what it's saying here. So what's the next one? Again, you have to know that we lose G and gain F. If we're losing F, then we're gaining G. Again, the best answer. This is a good question. This is a very good question. You guys probably got this one wrong. But it shows you, right? It literally shows you how the MCAT is, how you have to pick the best answer. This one is pseudo discrete. You should know this. You should know this as well. So yeah, that's pretty much it for this video, guys. So when you're reading, read slow. You know, realize that it's not that complicated, these passages. This passage, I think, is a little easier. If you want me to go ahead, choose a passage if you guys already have this, if you bought this. Choose a passage, okay, and tell me to do it. I'll break it down for you guys. But when you guys are doing these passages, make sure you understand, like, listen, like, it's not that bad. It's easy. It's not easy, but it's doable. It's very, very doable, okay? This can get a little complicated here. These passages get a little complicated, but these questions, they're very, very doable. Look at this question, like, which type of enzyme is linked with, to suppress CBC? That's easy. Like, these questions are easy. Literally, okay, go to table 1. It literally tells you go to table 1. Boom. Like, these questions, they're not that bad. But this can be a little complicated, but these questions are not that bad. All right, this video got pretty long. Let me know in the comments if you want me to, you know, break down another passage. Which passage you want me to break down if it's a little complicated for you guys. Till next time, Erickmed here. Let me know what you guys think. Comment down below. Like, share, subscribe, do all that fun stuff. And I'll see you guys in the next video.